Hi guys, this is going to be the second video of the cell cycle and cell division chapter, right? And in the first part, I talked about the basics, why there is a need for cell to divide, what kind of a division do we have, right? So in this lesson, we are going to talk about the interphase, right? So let's begin. So when a cell divide, right, we have mainly two phases. The first phase is your M phase, and then the second one is your interphase, right? So mainly we'll start with the interphase, then we'll move to the M phase. So what is interphase? Interphase is the longest phase of the cell cycle, and this is the most active phase. Why? Because all the requirements of all the requirements of these the cell division, right, so is being done in the interphase. So this is going to be the most active phase where all the requirements, all the protein synthesis, RNA synthesis is being done, and this is going to be the 90 to 95% duration of the cell. Means the interphase is going to take what, 90 to 95% duration of the cell. So suppose, let's say, the human cell, divide in 24 hours, right? So this, out of this 24 hours, right, so this 90 to 95% duration of this, the cell will remain in the interphase, right? Okay. So the first phase of the interphase is the G1 phase, right, or the first gap phase. In this case, in the G1 phase or the first gap phase, most of these cell organelles of the cell remain, it will increase. Why? Because we need to synthesis for the growth factors. We need uh, the synthesis of the nutrient, right? As well as we need the protein synthesis, protein and RNA synthesis. That is what going to be required for the next phase, that is the S phase, right? And here the cell grows maximum in the G1 phase. Okay, now let's talk about the S phase. In the S phase, that is known as what, synthesis phase, where the DNA synthesis is being done. Okay, so here in the G1 phase, what we saw, all the growth factor or all the proteins that was being synthesized will be utilized here in the S phase for the DNA synthesis. Okay, so here along with the DNA synthesis, histone protein is also being synthesized. Histone protein is basically what, uh, it is helping the DNA is what, wrapping up. Means if this is the histone protein and this is the DNA, right? So histone protein will wrap around the DNA, and ultimately you have thin thread like a structure. It will be what, it will give you what, chromosome. Okay, so this what, condensation, compactness, right? So compactness is what is being done with the help of the histone protein. Okay, so if I tell you what kind of histone protein do we have the H2A, we have the H2B, we have the H3, and we have the H4 molecule, right? So we have two molecules of the H2A, we have two molecule of the H2B, we have two molecule of the H3, and we have two molecule of the H4. All they come together and form what, octamer, histone octamer, right? So this is the histone octamer. Okay, and this is the DNA. This is the DNA. So DNA is going to wrap up around the histone protein or that histone octamer. This is the another histone octamer, and this is what give you the nucleosome, then solenoid, and ultimately you will get the chromosome. Okay, so for more about these, I'll give a link. You can watch the difference between the chromatin, chromatid, and the chromosome. Here you will find, what is the exact difference between these two. Okay, now let's talk about the S phase. Okay, what is happening in the S phase? Simply the DNA synthesis as well as the histone protein synthesis, right? But the most important thing is here, there is no increase in the chromosome number. All right, but if you talk about the DNA content, the DNA content is what, getting doubled. What do I mean by this? See, when a cell is there in the S phase, when a cell is there in the G1 phase, right? So in the G1 phase, you have a DNA something like this. But when a cell enter into S phase, so this portion, this thing, that is the chromosome, it is what getting copied, and you get something like this. So initially, this portion was your this original DNA, and this one is what, the that is thing we this thing is being synthesized in the S phase. So that's what the DNA cutting is getting doubled, but here the chromosome is one, here the chromosome is one, right? So that's what the DNA content is getting doubled, but the chromosome remains the one. Centriole replicate in the S phase, right? So this simply means what, the centriole is going to what, replicate in this phase. Centriole is basically what, going to give the spindle fiber. So spindle fibers is going to what, attach to the kinetochore. So this centriole is playing very important role here. Okay, now let's talk about the G2 phase. So gap 2 phase, and this is what, the final preparation of the what, this is the what, final preparation phase, where the what, the M phase, where the actual division is being done. So all the preparation, the final preparation is being done in the gap 2 phase, right? So if this is the cell cycle and this is the M phase, G1 phase, S phase, and the G2 phase. So here the preparation is being done for the S phase. Here in the S phase, DNA synthesized. Now in the G2 phase, what is going to happen? We need all those material that is going to be required for the M phase. So that thing will be done here in the G2 phase. So all the material required for the M phase is being synthesized in the G2 phase. Specifically, the important one is the tubulin protein. So tubulin protein is what making your spindle fibers, right? And that spindle fibers is what going to attach to your kinetochore of the chromosome, right? Fine. And then after all these things is being done, then the cell will move to your M phase. Sometimes, okay, sometimes cell remain in the G naught phase. G naught phase is also known as what? Gap zero phase. Gap zero phase or the quiescent stage. Why? Because see, suppose you have some cells, some cells just like your heart cell or the nerve cell, right? They are what? Fixed in number. They do not divide throughout their life. Why? Because they are there in the G naught stage. Okay, so in the G naught stage, cell remain metabolically active, means all those metabolic reaction like the catabolic reaction or the anabolic reaction, the glycolysis, the TCA cycle and all those reaction will go on. But the thing is, cell is not going to divide from one cell into two cell. So that's what not happening in the G naught phase. Cell is metabolically active, but it's not going to divide. So that's what the G naught phase. It's what kind of what the resting stage you can say, where the simply cell is active, but it's not dividing itself. Now, after the division, we have some other phases like the checkpoints, we can say. Okay, so why checkpoint is there? Simply just to check whether in the specific stages like the G1 phase, what the role of the G1 phase, where the nutrients is being synthesized, growth factor is there or not, the DNA damage or not. Means all those things, if there is a DNA damage or if the growth factor is not synthesized in the G1 phase, or the nutrient which is what required for the S phase is not there, then the cell will be what halted in the G1 phase. Cell won't allow to go for the S phase. That's what the actual role of the checkpoints. So it is what checking at the specific stage, and it is what checking the requirements are there fulfilled or not. So here, if you'll see, we have got the G1 phase, then you got the S phase, then you got the G2 phase. And between the G1 phase and the S phase, there is one checkpoint. Okay, and that checkpoint is what checking what the nutrients are there or not, growth factors are synthesized or not, if there is a DNA damage or not. If any of these is what absent, like if there is a DNA damage, cell won't allow to go for the S phase. Okay, so what is happening in the S phase? DNA synthesis. If the DNA is properly synthesized or not, DNA is properly replicated or not. So if that thing is not done, then there is another checkpoint, and that's what cell is halted here. Okay, then in the G2 phase, just after G2 phase and between the M phase, we have another kind of a checkpoint that basically check the cell size and the DNA replication is being done properly or not. If there is some kind of a mutation or if there is some kind of error mismatch, so cell won't allow to go for the M phase, metaphase. And in the specifically in the metaphase, there is a mitosis phase. There is one specific stage that is known as what metaphase checkpoint, where the kinetochore, at the kinetochore, spindle fibers attached. So if that spindle fiber is not attached properly to the kinetochore, means the separation of the chromosomes or the chromatid is not going to take place properly. So this is very, very, very important, right? So for this we have a specific checkpoint in what the metaphase. Okay, so what is checkpoint? So checkpoint is specifically what? Proteins. What? Proteins. So we have the proteins. So we have two kind of a protein. The one is the cyclin protein. So we have the cyclin A, B, C, D, and all. And along with this, we have the another kind of a protein known as what? CDKs, cyclin dependent kinases, right? So CDK2, right? CDK4, CDK1. So all they come together and they are acting as a checkpoint. Okay, so as you can see in this image, there are various kind of what? Cyclin E, cyclin CDK, CDK4, okay, so all these are what acting as a checkpoint. All right. Okay, now let's talk about the M phase. M phase is basically known as what? Mitosis, where what is happening? The nucleus is dividing plus the cytoplasm is going to divide, right? So for the nucleus division, we have a specific term known as what? Karyokinesis. Karyo simply means what? Nucleus, and kinesis term simply means what? Division. Okay, so cytokinesis simply means what? Cytoplasm, and kinesis simply means what? Division. So cytoplasm division is going to take place. All right, so M phase, mitotic phase, Karyokinesis, nucleus division, cytokinesis, cytoplasm division. And the karyokinesis is further divided into subphases. The phases are prophase, metaphase, anaphase, and the telophase. So prophase, metaphase, anaphase, and telophase, they all come together and known what? Karyokinesis. Okay. So M phase is the what? Mitosis or the mitotic division. This M phase or the mitotic or the mitosis basically occur in the somatic cells. What is somatic cells? So all cells of your body, all cells of we basically based on the division, we should basically classify the cell into two categories. The first category is the somatic cell, and then second one is the germ cell. So germ cells are the cells which give rise to what? The ovum, the sperm, gametes, right? So apart from the germ cells, all cells are your somatic cells, right? So in those somatic cells, M phase is going to occur, and this kind of a division is known as what? Equational division. What do I mean by the equational? See, initially, suppose you have the two chromosome here, right? Suppose, let's say this is the skin cell, right? And when this cell, it go for the cell cycle and cell division, and at the end you get the two cell from it. And the how many chromosomes are there? Equal number of chromosomes. So you got the two chromosome here you got the two chromosome, here you two chromosome, and they are your daughter cells. So in the daughter cell you got the equal number of chromosomes. So that's why this kind of a division, M phase, is known as what? Equational division. Okay, so let's talk about the karyokinesis. And karyokinesis, the first stage of the karyokinesis is the prophase, and this is going to be the longest phase of the mitosis. Okay, and here the chromatin fiber are entangled and condensed. So initially they are what? Entangled, and now they are going to what? They are going to what? Go towards the more complex stage, that is the condensation towards. Okay, so as you can see in this image, prophase one, this is the early prophase. Here the condensation of the chromatin begins. What do I mean by the chromatin? See, this is the what? Initially thin thread like a structure is what? Your chromatin. And when in the what? During the condensation begins, right? This chromatin takes place what? Shape more complex. So that's what your chromosome is. So initially what? Thin thread like a structure is your chromatin, and then the condensed form of this is known as what? Chromosome. What do I mean by the condensed form? See, I just explained you that this is your DNA, and this DNA get wrapped up around the histone protein, right? DNA wrapped up around the histone proteins, and this gives what? Complex structures like the nucleosome, like the solenoid models, or these are what ultimately give rise to the chromosome. So that's what the condensation of the chromatin fibers begins, and the cytoplasm become viscous. In the late prophase, what is going to happen? Chromosomes condense properly, right? And then chromosome begins to move toward the equator. Okay, so suppose you have the some chromosome here, so they are going to what? This is what? Equator. So they are what? Going to move towards the this side. Then nuclear membrane starts to disappear. Nuclear membrane, as nucleus is double membrane, so they are what? About to disappear. Along with this, organelles just like your ER, the Golgi, vacuoles, lysosomes, other organelles, they're what? About to disappear. And then the centriole will move toward the opposite pole. So suppose this is the nucleus, and here you got the this. So this is what going to move toward the opposite pole here. Okay. Now in the metaphase, what is going to happen? Now the nuclear envelope completely disintegrates, disappear. Now the chromosome condensation complete, means from the thin thread like a structure, you are with this. So you got this structure. Okay, now stage, the morphology of the chromosome can be studied here. So this is important line, so morphology, how the chromosome look like. So this stage is what best suits to what? For the study of the morphology of the chromosome. And the chromosome lie at the equator. Now in the metaphase, as you can see, this is the equator, right? This is the equator where all the chromosomes are aligned at the equator. And here what you see from the what? Centriole, the spindle fibers are coming, and it is what? Going to attach to your kinetochore of the chromosome. Okay, so chromosomes, what is going to happen in the metaphase? All the nuclear membrane completely what? Disappear, and here the chromosomes are at the equator, as well as what? The best stage to study the morphology. Okay, and this stage, I mean that equator where all the chromosomes are aligned, known as what? Metaphasic plate. Known as what? Metaphasic plate. All right. Okay, the another important thing, as I told you, that this is the centriole, and suppose this is the what? Equator. So this centriole is what? Giving rise to what? Spindle fibers, and that spindle fiber is what? Attached to your kinetochore, right? So that part, that part from where the spindle fiber is coming, that part is known as what, plus end, and the another end is known as what, minus end, and that minus end is what basically attaching towards your kinetochore, right? So plus end of the spindle fiber will be toward the centriole, okay, whereas the minus end of the spindle fiber is what, toward the what, kinetochore of the chromosomes. And as I already told you, that spindle fibers are what, made up of the tubulin protein, and these are being synthesized in the G2 phase. Okay, the next stage is the anaphase, and in this stage what is going to happen, the chromatids separate. So initially this is the what, metaphasic plate, phasic plate, where all the chromosomes are what, aligned, right? Now the spindle fibers, they come and got attached to the kinetochore of the chromosome. Chromatids is going to separate. Why? Because this spindle fiber will be pulled towards this side of the centriole, and this spindle fiber will be pulled toward this. So this leads to what, the separation of the what, the duplicated chromosomes, duplicated chromatids. So in the S phase we were having this, and after the S phase we got this, so we need to separate this out, right? So that's what happening in the NFL, right? So chromosome with the one chromatid move to the opposite pole, and this, the contraction of the spindle fiber lead to the movement of the chromatids to the equator, from the equator to the opposite pole. So as the spindle fiber is moving toward the this centriole, and this leads towards the separation of the chromatid, and this anaphase is the shortest stage of the mitosis. Okay, the next stage is the telophase, and this stage is exactly the reverse of the prophase. So what we learned in the prophase, simply the disappearance of the nuclear membrane, the organelles and all. Okay, so some same thing, just opposite, this vice versa of it is going to happen in the prophase, means the spindle fibers is going to disappear, the organelles is going to reappear, and the nuclear membrane or the nuclear envelope is what, going to what, reappear. Okay, and then the chromosome, which was, I mean, in this form, is going to what, getting converted into this chromatin fiber. Okay, fine. So we just completed with the karyokinesis, where the nucleus got divided. Now it's turn to discuss about the cytokinesis, where the cytoplasm is going to divide. So cytoplasm can be divided into two form, right? Where what is going to happen, the if we have the animal cell, as you can see in this image, so this leads to what, animal cell. In the animal cell, the cytoplasm division is what, from the periphery toward the center, and ultimately you are left with the two cell. So this is what, one cell, this is the another cell. Okay, such kind of cytoplasmic division is known as what, centripetal order, means where the contraction of the formation is what, there. Whereas if I talk about the cytokinesis in the plant, it is what, different. This kind of, I mean, the cytokinesis in the plant is known as what, centrifugal order, where what happens, we have the, if this is our nucleus, and as this is the double membrane, we have the ER, and then we have the Golgi bodies. So Golgi bodies, what Golgi bodies do, they release vesicles. The vesicles, these are the vesicles. These vesicles are filled with the magnesium and calcium pectate. So these vesicles, they will what, they will go and what, they will what, land there, I mean, land at the equator, and what, more and more vesicles, they are what, they will be what, there at the equator, and they will form the plate, the cell plate, right? And ultimately this will what, lead to the what, for what, division of the cytokinesis. So this thing, the vesicles filled with the calcium and magnesium pectate is what giving rise to the what, middle lamella. So what, so this middle lamella here and here. Okay, so this what, vesicles, they all will get fused, and this is what your middle lamella, and ultimately you are done with the what, two cells. So here the cytokinesis is being what, done, where what, the Golgi bodies is what, secreting some kind of vesicles filled with the calcium and magnesium pectate. They what, they go and land at the equator, or the center portion of the cell, and ultimately the cell plate formation takes place, and hence the cell divided into two. So as you can see in this image, in this, initially the vesicles are 1 or 2, now you have the more number, and ultimately you are done with the two daughter cells. Okay, so this is what the summary of what we just discussed in the simple, the division of the mitosis. Okay, now let's talk about the significance of the mitosis. Mitosis is so much important? Simply, as I already told you, that mitosis is going to take place in the somatic cell. Okay, and these somatics, most portion of our body is basically what is made up of somatic cells, and we need what these such kind of a division, the mitotic division or the mitosis. It is what required for the cell repair and the cell what replacement. Okay, and it is also required for the body growth. And this mitosis, it what restore the nucleocytoplasmic ratio. So as I told you in the first lecture, that when there is a what change in the nucleus and the cytoplasm ratio, this leads to what give the signal to a cell. Yeah, it's time to go for the division. Now let's go and divide. So it restore this nucleocytoplasmic ratio. Okay, and here the most important act, the mitosis, it produce the diploid cell, and that is what with the identical genome. I hope you have understood this, all the metaphase and all. If you have any doubt, any query related to it, you can ask me in the comment section. Thank you.